So now I will introduce Dr. Jordan Buck. Dr. Buck earned his dual PhD in neuroscience and integrative physiology with a certificate in behavioral genetics from the University of Colorado Boulder in May 2020. His dissertation research was conducted in the Stitzel Molecular Neurogenetics Laboratory and characterized the intergenerational transmission of neurodevelopmental disorder-like phenotypes conferred by developmental nicotine exposure in adolescent mice. Thanks, Jordan.
Thanks, Rose. So the title for this presentation is Developmental Nicotine Exposure Induces Intergenerational Transmission of an Ensemble of Neurodevelopmental Disorder-like Phenotypes in Adolescent Mice. So I'll start with a brief outline of the talk. I'll begin with some background and significance. I'll then move to describing the behavioral consequences of developmental nicotine exposure and the treatment thereof. I'll then discuss the neuropharmacological, neurobiological, epigenetic, and neurotranscriptomic impacts of D&E. And lastly, I'll close with some conclusions, implications, and future directions. So obviously, as you can see on this outline, this is a lot of topics. I've elected for this presentation to, instead of going on a deep dive into any one of these areas, I'll do a broad or bird's eye view of all of these findings, just to give everyone a broad picture of all the research that's been conducted as part of this project. So with that, moving to the background and significance, as we've already heard before, smoking during pregnancy is prevalent. It's also very costly, and it carries numerous public health consequences, including those for the mother who smokes during pregnancy, as well as her fetus. Most importantly, smoking during pregnancy is associated with a variety of neurodevelopmental disorders, including, as we've heard already today, ADHD and autism, and to a lesser extent, schizophrenia. So with that in mind, I'll move to our first set of data, which regards the behavioral consequences of D&E. So first, I'll introduce you to our mouse model, which is essentially an attempt to recapitulate this panel here on the left, where we have a mother who is elected to smoke during pregnancy. And in doing so, she is simultaneously exposing 3 generations to nicotine, namely herself, the F0 generation, her unborn child, the F1 generation, and the germline of that unborn child, which ultimately translates to the F2 generation. So we've used a very similar mouse model to that described by Dr. Beatty. I'll just briefly touch on this. So essentially, we use a nicotine and in a saccharin vehicle oral solution. That treatment is given beginning 30 days prior to mating to a drug-naive male, continues through weaning of the offspring. And for this reason, the F2 generation is a multi or intergenerational transmission rather than transgenerational, because the exposure continues through weaning, which we elected to do because of this model's smoking during nursing after pregnancy. So the first set of results here, and I'll be showing you only summary data for this presentation, just to keep it brief and to allow me to describe these different topics. So the main takeaways from our behavioral experiments are that both first generation D&E mice, shown in blue throughout the talk, and second generation, shown in green throughout the talk, exhibit active and inactive phase hyperactivity, which extends to a novel or open field environment. They also exhibit increased risk-taking behaviors, accompanied by increased voluntary nicotine consumption and preference. In addition, both groups of D&E mice exhibit alterations in the rhythmicity of home cage activity, such as late chronotype. And each of these phenotypes, in one way or another, recapitulate a symptomological characteristic of neurodevelopmental disorders such as ADHD or autism. And so ultimately, these behavioral findings suggest that developmental nicotine exposure in our mouse model does confer neurodevelopmental disorder-like behavioral phenotypes that are transmitted to the second generation D&E offspring. Next, we decided to look at the ability to treat those behavioral alterations. And so firstly, methylphenidate is a frontline treatment for neurodevelopmental disorders, particularly ADHD. And so we administered it to both first and second generation D&E mice, and both groups exhibited a therapeutic-like effect of methylphenidate on hyperactivity and risk-taking behaviors, as well as alterations in the rhythmicity of activity. Moreover, we administered nicotine similarly to these mice, and we found that consistent with the self-medication hypothesis for the increased use of nicotine in neurodevelopmental disorder patients, both first and second generation D&E mice exhibit a normalizing effect of nicotine on these same behavioral measures. So the hyperactivity, circadian alterations, and risk-taking behaviors. So summarily, both methylphenidate treatment and nicotine intake did exhibit normalizing effects on the behavioral anomalies induced in first and second generation D&E mice. Moving next to the neuropharmacological impacts of D&E. I'll first point out that corticostriatothalamic dysfunction is known to mediate hyperactivity, inattention, and impulsivity, as well as other symptoms in neurodevelopmental disordered patients, as well as D&E children in general. With this in mind, and given that nicotine acts through nicotinic acetylcholine receptors, obviously, we assessed nicotinic acetylcholine receptor expression and function, as well as dopamine transporter function in the frontal cortices strata and thalami. So keeping it brief here, we found that both first and second generation D&E mice exhibit alterations in nicotinic acetylcholine receptor expression, or binding more specifically, in the frontal cortices and strata. They also exhibit various alterations in the function of high and low sensitivity NACHRs in both the frontal cortices and strata. And that actually extends to the thalami as well. Lastly, we also identified impaired dopamine transporter function in both the frontal cortices and strata of both groups of D&E mice. And so one thing I'd like to pause and point out here is that you've probably picked up on the fact that almost all of these phenotypes that I'm mentioning are exhibited by both the first and second generation D&E mice. That's partly because I'm cherry picking and describing only the alterations that were multi-generational or intergenerational. However, on a broad basis, most of these phenotypes were detected across both generations. So just keep that in mind as we move through the talk. Next, we examined the neurobiological impacts of D&E. So I'll give a little bit of background here just to refresh everyone on these topics. So first, I'd like to point out that there's a delicate balance of pro-BDNF to mature BDNF within the developing brain. So as we all know, BDNF is first synthesized in a pre-pro form, which is then converted to a pro-BDNF form, which then has to undergo proteolytic cleavage to yield mature BDNF, which acts as we all are familiar with. Importantly, there are particular metalloproteases such as furin, which catalyzes proteolytic cleavage of pro to mature BDNF. And given this, we can imagine a scenario where if there were alterations in the expression of these proteolytic enzymes, that may perturb this pro-BDNF versus BDNF balance, which given the divergent impacts of pro versus mature BDNF may have substantial consequences for neurodevelopment. The other bit of background info here regards the HPA axis, which is a third order neuroendocrine circuit that uses the effector molecule glucocorticoids, that is cortisol in humans and corticosterone in rats and mice. So I'll point out here that glucocorticoid receptors exist in an inactive state in the cytosol. Given that glucocorticoids are steroid hormones, they diffuse across the cell membrane, enter the cytosol where they can bind these receptors, which upon activation will translocate to the nucleus and then ultimately modulate downstream gene expression when they act at glucocorticoid response elements or other DNA sites. And so there's also another important point here, which is that there are chaperone proteins such as FKBP5, which can bind and sequester glucocorticoid receptors in an inactive state in the cytosol. And so this is a regulatory mechanism for glucocorticoid receptor activation. I'll get to this all more in a second, but that's just some general background info to keep in mind. So we analyzed both the HPA axis and pro-BDNF to mature BDNF processing in both groups of D&E mice. So the important panels on this slide are these two on the right. So on the top, we have a hypothetical normal cell from our vehicle exposed or control mice, specifically from the frontal cortices and strata. And down below, we have our findings in both first and second generation D&E mice in those same brain regions. So the main things you can pick up on here are that on the bottom of the panel, there is an accumulation of pro-BDNF and a relative deficit of mature BDNF in both groups of D&E mice relative to our vehicle controls. And this corresponded to a deficit in furin expression in these mice. And so these data suggest that downregulation of furin expression may hinder the normal processing of pro to mature BDNF, and thereby explain the accumulation of pro-BDNF and deficit of mature BDNF that we identified in both groups of D&E mice. Looking at the HPA axis results, we found that Again, in both the frontal cortices and striata, both first and second generation D&E mice exhibit an accumulation of glucocorticoid receptors in the cytosol and a relative deficit of them in the nucleus. And this is suggestive of glucocorticoid receptor hypoactivation. So you have more in an inactive state in the cytosol versus in an activated state in the nucleus. Given these results, we looked at FKBP5 levels, that chaperone protein that can sequester these GRs in the cytosol. However, we found no difference in either D&E group compared to control for FKBP5 expression. So this suggests that FKBP5 does not mediate this glucocorticoid receptor hypoactivity in our D&E mice. However, what seems to be a more logical explanation given our data is that both groups of D&E mice exhibit hypocorticosterolemia, or a deficit in glucocorticoid content, relative to our vehicle control. So it seems that the GR hypoactivity is likely or largely mediated by the deficit in corticosterone levels in first and second generation D&E mice. So moving on to our next section, we wondered whether some of these neurobiological alterations could be epigenetically mediated. And so next we did some experiments to that end. As you all know, I would just remind you that both DNA methylation and histone modifications are the two main aspects to epigenetic regulation of gene expression. The important areas of this, for the purposes of this talk, are shown on this slide. So on the left, we have an epigenetically disinhibited gene site. So there is no methylation of CGs. There's no methylated DNA binding proteins bound to those CGs, and therefore no histone modifiers recruited to those sites. And so this results, again, in epigenetic disinhibition. However, if you have DNA methyltransferases that come along and methylate various CG sites, that will then facilitate the binding of methylated DNA binding proteins, such as MECP2, which can then recruit histone modifiers like histone deacetylase 2 to ultimately facilitate heterochromatization and the epigenetic silencing of gene expression. And so given this, we examined several of these factors, largely through Western blot analysis. And I'll begin by showing you the summary of those results. So again, as was the case in previous slides, on the top we have our control group for comparison, and below represents a hypothetical cell in the frontal cortices of either first or second generation D&E mice. So our findings herein were that both first and second generation D&E mice exhibit global DNA hypomethylation in the frontal cortices, also the striata. In addition, there are deficits in DNA methyltransferase 3A expression in both groups, which may explain that global DNA hypomethylation. Separately, but coincidentally with these findings, we detected deficits in both MECP2 and HDAC2 levels. So collectively, we can imagine, compared to our vehicle control group on top, how there is a general likelihood for an epigenetic disinhibition of the expression of various genes in our D&E mice because of the lack of DNA methylation, as well as the deficits in MECP2 and HDAC2 levels. Ultimately, that could result in a situation where there are more genes being expressed, or to a greater extent than would normally be the case or was the case in our control group. So these are very broad findings that just generally suggest that there may be epigenetic underlayment of the other neurobiological changes that I mentioned in previous slides. So for our last section of data, and thank you for bearing with me through the very fast talking, I'll point out some of our studies on the neurotranscriptomic impacts of DNA. And so we wanted to do a cell type specific transcriptomic analysis, and in doing so, we looked at our various options to that end. We settled on D1 type striatal medium spiny neurons because they're implicated in neurodevelopmental disorders. They're impacted by nicotine, and most importantly, perhaps, they have been very much understudied in neurodevelopmental disorders relative to other neuronal populations. So we utilized a cell type specific backtrap mouse line that allowed us to isolate DRD1 type medium spiny neurons from the striata, and then performed transcriptomic analyses through RNA sequencing. Here is a plot of the general results. So both first and second generation D&E mice exhibit up and down regulation of around or more than 300 genes. And about half of these differentially expressed genes were shared across groups, both for the enriched transcripts and the depleted transcripts. a little bit more detail on some of the top hits from this analysis. These are the top shared upregulated transcripts across both groups. So the top one was neuropeptide S receptor 1. Other interesting hits were secretogranin 5. Very interestingly, we found a large increase in the abundance of FKBP5 transcripts, which if you remember back to the neurobiological data, we actually found no differences in FKBP5 levels in strata or frontal cortices. However, this is a cell type specific analysis, so maybe that explains why we're finding FKBP5 increases in D1 type MSNs, but not in the striatum as a whole. On the down-regulated side, the top shared depleted transcripts included PNPT1, ELOF1, which is a elongation factor. We've got a histone, so on and so forth. So a lot of interesting hits here. In order to better capture the overall nature of these changes in gene expression, we performed a functional annotation enrichment analysis. Again, I'm only showing you the data for the differentially expressed genes that were shared between both first and second generation D&E mice, which is about half on the whole. The top enriched functional annotations shared across groups included terms such as translocation, methylation, neuropeptide, spermidine biosynthesis, and chaperone. Very interestingly, spermidine biosynthesis, spermidines are a class of cellular polyamides that are known to regulate DNA methyltransferase expression, and they negatively regulate it. And so this is actually consistent with our findings of DNMT3A deficits, given that spermidines are overrepresented at the transcript level. And therefore that may mediate some of the repression of DNMT3A expression that we detected as presented in previous sections. So moving on to the depleted functional annotations, the top shared ones included RNA binding, mRNA processing and splicing, nucleus, as well as another interesting hit, metalloprotease, and lastly, chromosome. So you may begin to pick up on here how a lot of these annotations are consistent with some of the neurobiological data that was presented in previous slides. So that's nice to see. Lastly, we also performed a WGCNA analysis of the differentially expressed genes, again, that were shared between the first and second generation D&E mice. And just briefly, if we zoom in on this, this turquoise module, we performed a Go biological process annotation analysis for that module. And a lot of interesting terms came out of this, which included terms such as behavior, locomotory behavior, nucleosome and chromatin organization, DNA conformational change in packaging, gene expression, RNA splicing, another important one, proteolysis, neuropeptide signaling, so on and so forth. So these RNA-seq data very much coincide with and support the findings that were presented all the way from the behavioral alterations through the epigenetic changes and the various changes in neuroendocrine and neurotrophic signaling. So with that, I'll try to wrap it up, and thank you again for bearing with me. So ultimately, both first and second generation D&E mice in our studies exhibit active and inactive phase hyperactivity, which extends to a novel environment, where they also exhibit increased risk-taking behaviors, in addition to increased nicotine intake and preference, as well as alterations in the rhythmicity of home cage activity. And most of these behavioral alterations were responsive to methylphenidate treatment and nicotine consumption. On the neuropharmacological side, both groups generally exhibited altered nicotinic acetylcholine receptor binding and function to one extent or another, as well as impaired dopamine transporter function, impaired pro-BDNF proteolysis, resulting in that pro-BDNF BDNF imbalance, glucocorticoid receptor hypoactivity, which may be explained by corticosterone deficits, as well as a number of epigenetic changes involving global DNA hypomethylation, DNA methyltransferase deficits, MECP2 deficits. And then on the histone side, downregulation of histone deacetylase 2. And lastly, as we just saw, numerous alterations in the D1 medium spiny neuron transcriptome. And so next, I'd like to try to weave all of these findings together into some sort of presumptive mechanism that may mediate the intergenerational transmission of D&E-induced neurodevelopmental disorder-like phenotypes. So we can imagine a scenario where D&E results in intergenerational neurodevelopmental disorder-like epigenomic perturbations, including, again, the hypomethylation, MECP2 and HDAC2 changes, DNMT3A downregulation. So those epigenetic changes may in turn confer neurodevelopmental disorder like alterations in the transcriptome, such as in D1 MSNs. And these transcriptomic alterations could then in turn confer the various neuropharmacological, neurotrophic, and neuroendocrine changes, which we discussed, including the NACHR and dopamine transporter dysfunction, changes in pro-BNF proteolysis, and the HPA axis alterations. And then ultimately, these neurobiological changes could finally manifest as neurodevelopmental disorder like behavioral phenotypes, including, again, the hyperactivity, circadian changes, predisposition to nicotine consumption, and therapeutic responses to methylphenidate and nicotine. So I have no data that proves that this mechanism flows in the way that it's shown here, but it is plausible given the data that we're presented. Lastly, just point out a quick potential future direction. So along the lines of the previous slide, you can imagine a scenario where you have a typical cell, such as that from our vehicle control mice, where you have sufficient DNA methylation and MECP2 and HDAC2 expression to facilitate normative epigenetic silencing of expression of a particular microRNA, in this case, microRNA 24. And that microRNA is known to negatively regulate furin expression. And furin, again, is responsible for the proteolytic cleavage of pro to mature BDNF. And so in this scenario, the cell is working properly. You have sufficient epigenetic inhibition of miR24 expression, which results in sufficient disinhibition of furin expression, which then allows normative proteolytic cleavage of pro to mature BDNF, which then has obvious consequences for neurodevelopment. However, based on the data in both first and second generation D&E mice, it may be the case that epigenetic disinhibition of miR24 expression due to DNA hypomethylation and MECP2 and HDAC2 deficits could, again, disinhibit miR24 expression, which would lead to an overinhibition of furin expression. And that may explain, at least in part, the accumulation of pro-BDNF and deficit of mature BDNF due to impaired furin-mediated proteolytic cleavage. And so that's just a hypothetical example. I think it'd be a nice experiment to tackle in the near future. Moreover, if we were able to demonstrate that this was occurring, this would provide an easy treatment opportunity. So obviously we could generate an anti-miR24 oligonucleotide, which could potentially bind and target for degradation the overexpressed miR24 if it were detected, which then could normalize furin expression and thereby normalize BDNF processing. Again, this is purely hypothetical, but it's a nice future experiment to think about. So with that, I'd like to thank the NIH for funding, the Stitzel Lab for support and guidance, particularly Dr. Jerry Stitzel and Dr. Heidi O'Neill. I'd also like to thank the Erringer Lab and individuals including Mark Melnick, Spencer Huggett, Stephanie Quintana, and my various faculty advisors throughout this research.